All right. So I think we're going to start. Well, thank you all for joining us today, and this panel discussion on data challenges in translational research in the pharmaceutical and the biomedical organizations. My name is Abel Ureta-Vidal, and I'll be one of the. Oops, I'm trying to get the. I will be one of the experts in the panel. And joining me here today, there is Xose Fernandez, the Chief Data Officer of Institut Curie. Hi, Xose.
Hi. Hi, Abel. Hi, everybody.
And also Alexandre Passioukov from Pierre Fabre, the VP of Translational Medicine. Hi, Alexandre.
Good afternoon.
All right, thank you. Right, so if you can see that you have three people from three different areas, and Alexandre represents, I would say, the end user, the one using the data to kind of generate new discoveries and find new drugs and cure some diseases. Xose is Institut Curie, representing the academics in here, and also the hospital, because Institut Curie, as he talked to you about, has healthcare duties as well. And Xose, as a Chief Data Officer, is very much in charge of the data, as opposed to being in charge of the IT. And there's going to be maybe some discussion we can have in there. And myself, I'm representing a software vendor, building an enterprise software platform to enable the best use of the data and pinpointing the insights. So we're going to run the webinar as follows. We're going to have three presentations, so roughly 5 minutes each. We'll take our time if it's going to need a bit more. So I'll start with my presentation, tell you a bit about Eagle, and then Xose will take the hand, talk about Institut Curie and what they're doing over there. And then Alexandre will close the presentations. And then we'll move on into kind of a panel discussion and Q&A. The webinar is recorded, so for people who want to kind of listen to it and see it again afterwards, we'll provide the links. That's kind of, we'll send you that after the webinar by email. While we are presenting, you can obviously ask questions through the GoToMeeting kind of application, and have here Catherine with me, marketing manager, kind of assisting me on that. So please, during the presentation, just type and chat your questions so we can use them at the end. Right, so I'll start, and just kind of by giving you a bit of an update about what Eagle Genomics is. We are a software company, and we are based in Cambridge, UK, in the Wellcome Genome Campus. That's just next door to the Sanger Institute and the European Bioinformatics Institute. And we're a software company building an AI-augmented knowledge discovery platform for life sciences R&D specifically. And we've been working for the last 9 years, nearly 10 next year actually, in three main sectors: human and animal health, personal care and cosmeceuticals, and food and nutraceuticals. And the ambition of the company is to build a platform that will be the innovation platform for the genomic era. And we call it, in a bit of a kind of a provocative way, the automated data scientist. And this platform should enable true data-driven innovation and decision-taking, and also increase the success rate of innovation by capturing kind of innovation and insight through data. Just to give you kind of a bit of insight about the kind of customers we're trying to help here, and I take here the architect of what I call the end user, the biologist. Here is Jennifer, and I can talk about her because I'm a biologist myself by training. And in our worlds, we have a kind of tacit knowledge in our heads that is putting in context and connection different entities of interest, as we call them at Eagle, either patients or drugs or genes or pathways. And those connections are basically supported by evidences. You can call that being publications that you've read, but it can be experiments that you've generated internally in your company. And to be able to update this kind of network of concept connections in your head as a biologist, you have to go through quite kind of painful, sometimes interaction with many stakeholders in the organization. I put here two of them only, a biocurator or bioinformatician, that are helping Jennifer to transform what's on the right here, what we call data sources, very IT-ish format of the data, either from open data access or from experiments generated in the organization, a very, Excel spreadsheet, flat files, SQL databases, you name it. And what we're trying to do here with Eagle is transform the IT form of the data into a form that the biologist can inherently understand better. This kind of knowledge connection or knowledge graph, if you will. Now, to do that, we've been working with pharma industry quite a lot, but in other sectors as well, and mentioned that in the following slide. We cannot believe that for developing a translational medicine platform, this platform needs 4 main capabilities, each of them being independent and can be deployed independently. The first one is obviously data ingestion, curation, or enrichment. And again, we're talking about translational medicine, so here is around the patient and building a catalog of patient data that is well annotated and enriched. That's the first capability. The second one is once you have, let's imagine you have your 1 million, 2 million patient records with all the data attached around them, is how do you prioritize and select the cohort of patients or cohorts, if you want to build different groups of patients, that are relevant for the analysis you want to do. And when you have those cohorts, then it goes into the capability 3, where you do comparison analysis. You go to cohort A, you compare it with cohort B, and you want to understand what's the difference between the two, either genetic differences or molecular assay differences, any kind of characteristics that enables you to differentiate the two cohorts. And obviously, once you've done the analysis after capability 3, you want to have another capability that is the ability to visualize and pinpoint where are the new discoveries and new insight the analysis has been able to provide you. We believe that all those 4 capabilities can be deployed independently, and obviously there is questions about how they interoperate with each other. And I've put that in a bit of framework around translational medicine and the idea of going from the bench to the bedside. And you start with the patient, has medical history, lab results, biomolecular assays, imaging even more now. And I've put here the kind of the process that I described before, where you start creating the patient data, building the catalog, then being able to select the patients to build cohorts and do the comparison of cohorts and then pinpoint the insights. And all of those kind of processes are supporting different applications. And I put here a few: identifying therapeutic targets, maybe diagnostic biomarkers, understanding kind of the etiology and the biological mechanism behind the disease, maybe some drug repurposing. And all of that enables you then to kind of design your interventional studies, clinical trials, or maybe decide to have a more observational study, either retrospective or prospective. And that in 2 different settings, either internal, because data is only on your R&D organization, or maybe in a collaboration aspect. And obviously we use that network, that framework, sorry, to drive our product roadmap. And you can see some of the modules here that we've been developing against each of the capability. And as I said, these capabilities are very well defined and are discrete. So we can module the deployments and have even third party kind of doing one capability as opposed to another. And what's important is how do you interoperate between one and another. One aspect that I wanted to mention here is that's not only valuable for translational medicine research. We've been applying the same framework in other industries. Preclinical can be one that is looking at not patient data, but you're going to look at another kind of focus of interest. They're going to be the animal model, the mouse, the rats, or even the cell line. Okay, you will need this kind of process at the same time to be able to select your cell line and the building maybe kind of a different type of cohort of mouse or rat, for example. But we did that also for animal health. So working for an animal health company at the moment, looking at cows, for example. They're not patients, but they are animals, and it's the same kind of process. Even personal care and cosmetic companies, the focus of interest is not the patient, is the consumer. And the consumer always has to be defined by specific phenotypes and data around it. And the last one is agri-tech industry. The focus of interest is not going to be the patient this time, it's going to be the plants, and how do you use this platform or this framework to do plant breeding programs. Okay. And I'm going to finish with just one slide about what are kind of the 5 main hurdles or challenges we are seeing in all the customers we're being kind of talking to and working with, and also for our own development of the platform. one aspect I talked to you about the end user. The end user is very much the one who's going to be benefiting from the data. And the user experience of any kind of a tool, software you're going to be putting in place that is very specific to a business case is what is going to drive adoption. You can have the most clever backend optimization, database structure, graph structure. If the rendering to the end user is not kind of useful, it's going to be very hard to be used. The second one is interoperability and integration with existing systems. We sell enterprise software, and so we are going into companies that have already systems in place that have been there for 5, 10, 15 years. And we have to have the ability to understand the information architecture so we can deploy a platform that can work with those systems and not ignore them. The third is the use or even the potential creation of standards that are not there yet. And I'll put here an example about FAIR principle. That in my view is not a standard yet, but we need those kind of standards that can be implemented within the software to enable smooth data and insight data exchange. The fourth one, finding the relevant data is important, but we are going beyond that, and it is how do you represent the insight for systematic capture. And when I say insight, is data is used, is analyzed, the outcome of the analysis is insight, and we have to be able to represent that so we can share it across the organization or with collaborators. And that brings me to the last point, and I think Xose is going to mention that as well, is the idea of federated data governance and the involvement of cultural change. We believe that solving the translational research big data issue in the pharma industry and biomedical research organizations is just not a technology issue. It's also a cultural issue in terms of changing how we see data and how we use data. So I finish with that, and I guess some of those points can be discussed at the Q&A afterwards. And I'll pass on to Xose to kind of talk about the Institut Curie and what he's doing there.
Thank you very much, Abel. That beautiful photo of Kingston made me feel a bit nostalgic, but the view, the view from my office, so I'm not jealous. I have the Eiffel Tower, and it's a bit foggy today, but it's still beautiful. And yes, we are. I'm going to be telling you a little bit about how we are tackling these problems here at Curie in Paris. And for those of you who are not familiar with Institut Curie, this is a private foundation which was created in 1909. It has 3 missions. You mentioned the hospital care, but we have also a strong component of research and also disseminating knowledge and preserving the Curie legacy. And one of the goals that we have in my hands is that from my hands, one of the missions of our organization is from the around 1,000 new cancers which are diagnosed every day in France, trying to make the most of them, joining those 10 million people in Europe who are survivors of cancer. And that means that we are working with a team of people from various nationalities. We have people from around 79 countries. We are around 3,400 people, and we are looking after around 10,000 new cancer patients every day. And the situation right now, it's we are in a space where computer power is not the bottleneck anymore. And we are on the next slide, Abel. We are, we can analyze and process huge amounts of data, and a combination of both the pressure from the increasing costs of healthcare, as it happens in Europe, alongside demands from society want to empower patients, are the driving forces for this transformation of services. And diverse data sets are being integrated and aggregated, allowing us to explore new interactions. The whole idea of this approach is to deliver a digital ecosystem. And although I've mentioned that we are based in France, we are working at the European level to make this happen. But amongst the challenges that we are facing is not only facilitating the creation of data, most importantly, increasing its access, use, and reuse to make the most of these resources. And as you've mentioned, also we are guided by the FAIR principle, so we want to make data findable, accessible, interoperable, and reusable. Our data management plan promotes data sharing, and we support our data creators to submit the data to sustainable repositories, and some of them are just across the road where you are based right now. And we are also advocating metadata as the way to contextualize data sets which enable this aggregation of data. I will show you later some examples of data integration which are used in some of the tools that we are having developed either at Curie or we are part of consortia using this. And I will also mention some of the projects that we have in the pipeline. If you show the next slide, we can see the kind of diverse data collections we have. We have, whoops, the previous one. No, the next one, yes. No, previous, that one. We have, the population, we have around 460,000, almost the population of Luxembourg in terms of patients for which we have information on treatments, on medical acts. Our, resources, our historical health records are handling about over 10 million documents. Every day, 3,000 new documents are added to our systems, as well as 1,300 new scanned documents. We have millions of images, and this is increasing, 40% increase in the number of images that we are storing in our resources. And we also have a biobank at our disposal with around 2 million samples. So what we have is, in some cases, we are initiating or we're leading a clinical trial, and we need to identify a cohort of patients meeting certain criteria. And for this, we have tools like this console, which allows us to select these virtual cohorts. But we also have tools to visualize, and you can see down here at the bottom, having a longitudinal view of a given patient, where we have an image of all the different treatments and when that happened and which samples are available, whether there is DNA extractions, whether there are biopsies, and we have all this information structured, so we know whether this biopsy was taken before radiotherapy or before chemotherapy. How we are working and how we are aggregating this data, and this takes us to the next slide. So we are creating, as I've mentioned, a data ecosystem that, amongst other things, one of the goals is to improve the patient access to clinical trials. So we have selected a pilot. We found a team which was very collaborative, but they had a very diverse collection of data. They have genomic data, transcriptomic, obviously information about the patients, and they had to invest some time, and you also mentioned it in your part of the presentation, to improve the metadata. And we have been developing, identifying which are the different dimensions. So we added a semantic layer and complemented with this metadata, which has been added, putting us in a situation to create an infrastructure which is in line with the requirements of the new legislative framework that we're having in Europe as from May next year, the General Data Protection Regulation. And we handle not only links to raw data, but also information extracted from this, information obtained from these electronic health records. Finally, also summarizing a little bit some of the challenges that we face and I faced when I joined Curie as Chief Data Officer. Biomedical sciences are very data-intensive enterprises nowadays, and the scale is ramping up very, very fast. So one of the first goals was to set up a strong foundation and designing the data architecture as a critical step to establish a framework for using this data, but also in line with the current legislation and the future European framework, protecting confidentiality and controlling access to patient data. We are working on this fair space, so investing time in developing those metadata that allows us to aggregate data is critical. And particularly in organizations such as Curie, with large amounts of types of data. But we strive to make this data truly useful. And as when we came, we have to change a few things. And one of the things is that putting data at the core. Information is an asset, and we are translating this into information. And this is, in a nutshell, what we are doing here in Paris and what brought us here to Paris. Back to you.
Okay, thank you, Xose. Very, very good. Thank you very much. So now we go to Alexandre and put the next slide. Alexandre.
Yes. We can move directly to the next one. So actually, for those amongst you who don't know Pierre Fabre, definitely you know some of his brands, because actually it's pharmaceutical company number 3 in France, but also one of the world leader in dermocosmetics, you know, with Abel, Curie, and other famous brands. In terms of pharmaceuticals, actually, our key area is oncology, dermatology, and neuroscience. Next one. Before actually speaking about how we see the digital challenge in translational medicine today, I would like to say actually we have a global challenge of productivity of research and development in the pharma industry is a general problem, a long-term crisis. We already see that the return on investment is less and less attractive. So it's symptomatic of the general change in the way we generate the innovation, and maybe to position a small pharma or a mid-sized pharma like Pierre Fabre, but also the big pharma, it's important to see what is the general direction, where we are moving to what. And actually, this slide is very generic, but actually the vision would be, of course, that healthcare is going to be based on precision medicine approaches, which is going to be based on a common bioinformatical foundation. So that's something which looks quite evident. It's not a real, let's say, a prediction. It's happening very quickly. The same actually about the research. Research is moving to a global network. So we are more and more connected to other people, and we depend actually on the data generated by other people. So the next slide is going to show you actually how, in the framework of mid-sized pharma, I propose actually to address translational questions. Because the questions we have, it's how we select the patients in early clinical trials first to get better chances of success, to demonstrate the proof of concept, proof of mechanism in clinical trials. Later on, potentially, we can develop companion diagnostics based on this approach. But also, it's very important that we can propose new targets, new therapeutic targets, based on unmet medical needs, which means based on patient data. Speaking about the data, it's the left-hand side block, actually, what I call in silico platform. Today, we actually put together all the data from all sources around 3 therapeutic areas. So I just refresh oncology, dermatology, neuroscience. The idea would be actually to collapse all the data from patients, from clinical trials, inside the company, the data we got from the outside, but also preclinical piece, which means the molecular data from the preclinical models we use to test actually some early molecules or the combinations, even cell lines data, which is very important from translational perspective because we want to see how much our preclinical models represent actually the patient situation, and if it represents, so which segment exactly it is about. Importantly, actually, for any pharma, but especially for a mid-sized pharma, it's important to find some specific niches where actually we develop the therapeutics to. And one of the niches from my perspective actually is going to be interface between indication. So we're really looking into the interfaces between oncology and dermatology, or dermatology and central nervous system diseases. So it's one approach. Another one obviously is going to be to see the segment of patients with a major indication, which may represent kind of orphan disease within a big disease. So next one. So the key principle is going to be actually we need to use cutting-edge technology, and the digital technology is one of the most urgent needs actually, and which is underexploited in my view in the pharma industry in general. I put in place here a semi-virtual structure with a common database which is agnostic of therapeutic area. And obviously we are success-minded, so we want to have to increase probability of success of our molecules, and we synergize with the research. How we can help the research people, because actually today we can we don't wait maybe mice data or cellular data. We can directly propose them some targets coming from patient databases. The advantage of this approach, of course, is we already know which patients are going to be the customers in the future, so we can directly tailor the clinical trials to those customer patients, and to develop some tools to identify these patients in the clinical trials or even at the level of companion diagnostic. And as I mentioned already, so we can also help to create new niches in some indications where previously we don't have ones. And a couple of last slides. Definitely, the data, it's the pain points, because nobody has enough data in the end of the day. So here you see actually the building we are currently now in Toulouse. And so in terms of digital collaborations, we have already some partners close or far away, where we either just directly access to the data, so we just pay for service, and we make some analysis on the databases already generated by some of our partners, or we have some true collaborations where we actually generate the data ourselves. So finally, I would like actually to enumerate the key challenges as I see it from the inside of pharmaceutical industry, because previously I was working for the big pharma, now it's mid-size. One big issue is actually the lack of clear understanding what is going to be the business value of this type of exploration. So it's the very, very important one. The second one is the data access, data silos. I already mentioned nobody is rich enough, so everybody has some data we don't have. Even within the company, sometimes data are not really shared, and people don't have access to this data, which we call data silos. The third one is how we can navigate in this data lake, where everybody use different formats, standards, and dictionaries. And finally, weak signal detection to get competitive advantage. Definitely, and that's maybe the difference with the academia. We really want to be the first one to find some signal, which is today maybe not the mainstream, to make sure we have some competitive advantages, and we're the first one to develop some target, some combination, or even address some new indication. That's all from my side for the introduction.
Well, thank you very much, Alexandre. Thank you very much, Xose. Just kind of I had one comment from the audience about when we presented the kind of the framework for the data flow. That we should mention that obviously there should be there, ingestion of the open data in literature and potentially kind of even internal data. So there are two aspects here. One is obviously you can only do new discoveries in the context of what's known already. So sorry, that was implicit that when you start working on research, whatever research, you have a base of the knowledge that's out there, and what you're looking at is you're generating new experiments internally. Is what are those new experiments providing that is new as compared to what everybody knows, and that's kind of one of the key challenges when you are just not one scientist in your lab, but you are in an R&D organization where there is maybe 2,000 to 3,000 scientists. Is how do you kind of pinpoint and kind of identify what is the analysis of the data generated by those experiments that is providing innovation in the company. That's a good point. So I'll probably add in that to the slide. I have kind of few questions in the audience. So I'll start with one, maybe kind of putting kind of Xose a bit on the spot here about the GDPR. There's a question about GDPR and how is that potentially comparing with the one in the US, the HIPAA law in the US about patient data privacy. Do you have a look at that a bit in that sense, and what is adding that is not there in the US already?
I mean, the GDPR is more comprehensive because it's obviously, I mean, HIPAA is only limited to clinical data. GDPR is any data of EU citizens. One of the big differences is that GDPR is also applicable in the US and beyond Europe. So that's a main difference. And it is not about, HIPAA is a lot about how you manage and what kind of standards the actual systems to handle clinical data will have. So there is a strong component in terms of where you can store it, what kind of servers, what kind of services you can use around data. In the case of GDPR, the emphasis is more about ownership of the data. The data belongs to the individuals, to the patients, and therefore they have the right to ask any company to delete the data. And obviously, in our case, as a hospital, patients can come and say, I don't want to have my data into your hospital. So there is a balance between respecting the will, and basically that data will be removed and it's not going to be identifiable, but it's also a strong component in terms of healthcare and public, as a hospital, you cannot ignore data. It will have impact in other people. So you have to find that balance. But I think that the main difference between GDPR and HIPAA is on that HIPAA is just limited to clinical data and how it can be managed, whilst GDPR is a much more has wider implications.
Okay. Do you want to add any comment on that, Alexandre? Or?
Well, I mean that's a very difficult one, because indeed the access to the data and ownership of the data, it's a complex matter. But actually today we witnessed the appearance of new companies, kind of startup companies, which try to resolve this type of data access or data privacy issue. For example, I recently have an interaction with the company Shivum. It's from Basel, from Switzerland. So they offer this, genomic blockchain ecosystem proposal, where they really define who can have access to which type of data. They develop some approaches how they kind of which type of incentives they can provide to the people to share the genetic data, and how they could generate this common pool of genomic. It's mostly about genomic for the moment, but potentially it can be used for all other type of big data repository. So I believe it's going to move quickly, because it looks like, considering the pressure and the interest from different stakeholders, probably once the technical solution is mature enough, it's going to explode.
So I have a few questions here, kind of on the panel. So and that's mostly directed to Xose and Pierre. I have a comment on that one. Is how do you assess the quality of data that you kind of get into your system, and how do you wait kind of to influence the output? So
the question was about data that we put in Curie. I mean, the data.
How do you assess the quality of the data that you are getting?
Okay. I mean, data. It's we have. It depends. We have different kinds of data. We have data that comes directly. When you're dealing with clinical records, you have, for example, we have talking about chemotherapy. We get the dosage directly from the software, which it's connected to the devices just delivering the chemotherapy. Similarly, with radiotherapy, we have the exact dosage, how many, what kind of irradiation was, what protocol. All that information is taken directly from the devices. We have open text, which is the comments from the physicians, and that's obviously if the physician makes a mistake, that mistake is there because it's part of the record of that particular. So in terms of quality of data, and then we have omics data. So we have if an exome has been sequenced or a gene panel has been sequenced for a given patient, yes, we do the kind of standard processes to validate that the FASTQs are at what they are, and the SNPs are validated by Sanger sequencing. So it's a mix. We have data which we cannot doubt because comes directly from the devices. So it gets, and there is no break in the chain, and there is data which is open that then we validate. When it comes to databases, we have QC controls in place to make sure that when people typed or dumped that information or record that information from the medical records, it's correct.
So one comment I want to add on that question specifically, and I will come with one of the kind of mottos of the Eagle Genomics in terms that value of data is more important than quality of the data, and you can have low quality data that has high value, and that's one of the work we are working with Pierre Fabre in some of the data is how do you measure the value, the relevance of a patient data to a specific kind of research use. I'll give you one example to show you that if I'm an oncologist working in current cancer, and I have an hospital coming to me and say, I have the best 20,000 cohort of diabetic patients, high quality with biobanking, sample, genomics, proteomics, imaging, and so on. Even though this cohort of 20,000 diabetic patients is very high quality, as an oncologist in colon cancer is very low value to me. So you can see that quality is not all. Quality is one component of the value and the relevance of the data. And one of the kind of paradigm shifts we want to be putting in place in the company, I think there was a question about how do you format, ingest the data in an automatic way. That was one of the questions. Is we can basically go to do what we call quick and dirty curation, because the output of the quick and dirty curation will be a measure of the value. And once you know which data is valuable, then you can direct and focus your curation efforts that are often human expert driven to the specific data that you want to use, because you know how valuable and relevant it is for your research. So there's a lot of differences and complementarity between quality and value. And in my view, value is kind of the thing that's going to go first in that sense.
I mean, and also something which is key also when you're dealing with clinical data is context. So,
yeah.
It is in which context that data was captured.
Exactly.
Which is essential.
Yeah, I would like to confirm actually that the quality of data, it's one thing, but when we speak about the innovation, especially early innovation, we need the right data mostly. So that's and the quality assurance, it may be a trap, because quality assurance is very important for confirmatory study. So when it's already, we know we're moving exactly. But when we're looking for weak signal, we cannot really, if you really want the perfect quality, it may become a challenge. So you need to balance in between. And that's why approach Eagle Genomics offers, it's very attractive to us. And actually we are going to launch the pilot study with them. In this sense, it's very, very important with dirty, which is not perfect, to get some weak signals where potentially we can generate some innovative hypotheses. Another point is actually sometimes we simply don't have the data, and that's I should like to mention it, because if you, for example, we used a lot TCGA database for different purposes, but we know it's mostly it's stage 1, rarely stage 2 cancers. So how well it represents the metastatic disease, where actually our main focus lies, we don't know. And so it's a big question, and sometimes we simply don't have the right data sets. It's not about the quality, we simply don't have it. Or the question of resistance to some immunotherapies, for example. We all know that big pharma, they just sit on their data, they don't share it. And so generally, but they're also poor in a sense because they only have their data. So we still have, we don't have any tool actually to share this type of data in a win-win type of system, and nobody really proposed such a system, and we need to convince people how we can go to next level of innovation where everybody going to find some value in sharing this type of data. And the quality, actually maybe I'm optimistic enough, but I believe the quality is going improving. So it's already based on the fact we already omics technology are maturing, so the data are becoming better and better. So recently we had some, we were defining some molecular signature for some innovative drugs, and we asked whether RNA-seq is really very important, much better than just expression profile we generally use before with Affymetrix, for example, panel. And apparently it's not a problem. So people say it's typically robustness of the signatures are very much the same. So the quality of data, sometimes it's not really an issue.
There's another question about what did you mean by federated data governance? And I guess that comes to me, but I'll kind of maybe put Xose on the spot as well there. What I meant by federated data governance is we mentioned that kind of data is distributed or in silos. And I think I don't have any issues with silos and departments that are generating data on their own. as far as they know how they need to share the data, what is the standard format, and what is basically the receiving end, the end users of organization want to receive. The problem with silos is when they are kind of put in place and there is no kind of data governance and same principles or standards saying, well, you are here the department of proteomics in this organization and you are here the department of sequencing. It's fine that you generate the data for specific projects, but when it comes to sharing the data, here's how you need to kind of be able to share it. Here are the standards and the formats. Now, how you generate the data on your side in your silo, this is your problem, this is your technology. The only thing I want is you to be able to kind of give me the data in the format that I expect and is consistent across the R&D organization. And that's what I talk about federated as opposed to centralized kind of data governance, where you spread across the standards and the processes for data sharing, and then you let them implement those data sharing, given that the standard is known and agreed. And that's kind of quite important in terms of cultural change and in terms of data governance. It's not just a technology issue, it's also about communication, about what are kind of the duties of every department and silo to be able to share the value of the data they have in a way that is digestible and usable by the other departments. That's what I meant by federation data governance.
Yeah, I mean, for me, for example, the main challenge with silo data is access is not a problem because you can develop the semantic layers. The main challenge when you have silo data is the lack of metadata. In some cases, you have a storage space for a given project. All the data around that particular project is going to be shaped by the project. And if you want to aggregate that data with a different project, with a different focus, that's what you have. If you don't have consistency with metadata, is where it becomes very challenging to aggregate that data. So in terms of when I talk about aggregating data and the governance around that, is more about defining what will be the minimum standards in terms of metadata to define the different sets and making the generators of data, make them aware that clicking always default doesn't add any information to the system, and they might want to reuse that data and they will miss that. So the time that is invested in curating that data is time that you will save in future uses of that data.
And so in terms of then kind of, there's a question here about, I think it's mostly for you, Xose, in terms of, maybe for Pierre as well, in terms of, how easy, you say that, patient data is important to get, Pierre was saying it's very hard to get kind of data and you have to get collaborations, but once you have those patients, how easy it is for you to build a cohort of specific patients based on specific characteristics or phenotype or metrics?
I don't know if Pierre, do you want to go ahead, or I can tell you, I mean, we are involved in around 200 clinical trials right now, and basically we identify our patients, we have the tools, and we have the criteria, age, phenotype, biomarkers, which treatments they had followed before. We have the tools, we have a number of tools that are relying on natural language processing, because this data tends to be unstructured. This data is text, so we have our querying tools to extract that information, and we can get our cohorts set within an hour. So in a week we can have identified individuals for a clinical trial within our hospital and partner hospitals that we can query as well with our tools.
Yeah. Alexandre, do you want to kind of say something about the acquisition of data, patient data, either within the organization and with collaboration, and how easy or difficult that is?
Yes, absolutely. I mean, it's absolutely very, very difficult, because actually even the clinical trials where we are kind of partially involved, sometimes it's difficult to get the full data from our partners. Mm-hmm. The approach is currently we have either we just define biological question, with people who have the data, have the ownership of the data, and we have for the fee for service, agreement. We just find a way. They make some analysis. The disadvantage of this approach, of course, we don't control exactly the way they do it. We don't know their data, so we're not in full control. Another another option would be just to get access to full database. So it's something which currently we're exploring with some partners as well.
Okay.
We also have the kind of discussion with people who identify virtual population, within France, for example, to make some prediction, to develop some predictive models on efficacy of certain, for example, pathway inhibition by single agent on combinations, what we call in silico clinical trials. So we don't have a clear flavor of how it really works, how predictive it really is. It's quite labor intensive and expensive actually, but we have a couple of pilot projects in this sense as well.
Okay, fantastic. Right, so I realize kind of we're a bit running out of time, so it's been 45 minutes, and I'm sure people are very busy, and I can stay talking about many of these things for hours. Well, I'd like to thank you, Xose and Alexandre, for joining me in this discussion here. And thank you for the panel, kind of the delegates who joined as well. As I mentioned, so we'll follow up with emails to kind of point to where the recording is. There are a few questions that have not been answered, and I'll follow up, personally on those ones individually, and help maybe find an answer if there is any. But well, thank you very much, Xose. Thank you, Alexandre, and in touch.
It's been a pleasure. Thank you.
Thank you.
Thank you very much.
Bye.
Bye.